Hi, this is Sadashiva Pai from Science Mission. Uh, Today we have Dr. Gupta with us, Dr. Vineet Gupta. So he is the of Vice Chair for Research and Innovation. He is also the Director of Drug Discovery Center. Dr. Vineet Gupta he is in the Rush Medical College in Chicago, Illinois. So Dr. Gupta has published several articles in various peer-reviewed journals, and he has received several grants and is in the editorial board of several journals. So welcome, Dr. Gupta, to Science Hangout.
Thank you, Sadashiv. Appreciate your
Uh,
inviting us here.
Thanks for joining us. Tell us about yourself, your background, your education, how you got here.
Yeah, good question. I have a fairly simplistic journey up to now. So I was
Okay.
born in India, did my undergraduate work in Delhi. I went to Delhi
Uh-huh.
University, and then I went to IIT Delhi, and then
Uh-huh.
subsequently came here for my graduate studies. I was at University of Rochester.
Uh,
I'm a chemist by training, so got my PhD in chemistry from there.
okay.
Worked with Eric Cool, so shout out to all the cool people.
Uh-huh.
Um, and after my PhD, I did a postdoc at Rockefeller in New York, and that's sort of the transition. That's where the transition in my mind happened from being a straight chemist to being somebody who's doing more biomedical translational work.
Okay.
So and since then, so I was at Rockefeller. After that, I dabbled in a couple of startups, got some taste of biotech world,
Uh-huh.
and decided that academia is a better suit for me.
Uh-huh.
So started. I was very lucky to get a position at Mass General in Boston, and
Okay.
then very quickly started my lab there, and have been sort of working in a similar field since then.
Okay. Tell us about your lab people currently.
Yeah, so I have a very estrogen positive lab, high
Okay.
estrogen lab. I have about 8 people in the lab,
Uh-huh.
6 women, 2 guys. So we certainly are in minority.
Okay.
Very interesting lab. People work very hard. People have a variety of backgrounds. I have 2 graduate students, 4 postdocs, a junior faculty, and a tech, and people from all over the world, people from US, people from Korea, from Asia, other
Mm-hmm.
parts of Asia, people from Europe. So keeps it very, very interesting.
Okay. Yeah, I know like people are motivated, these researchers are like, but how do you help them out? Because research goes and ups and down, has its own path. So how do you keep them motivated and get them going?
Yeah, very good question, Sadashiv. So research really is a marathon, not a sprint.
Yeah.
And so for marathon, I like, my mentors have prepared me. I have a very my philosophy is I let people learn at their own pace. I let them learn on their own. I let
Mm-hmm.
them make mistakes and learn from those mistakes. Mistakes are how you learn something. And also we have a very open system. My office door is always open, so people can come in and discuss anything, anytime, almost anytime.
Mm-hmm.
And we have a very positive lab, so very people help each other. Almost every project, there is a leader of the project, but there are several co-helpers supporting that leader. So that helps the lab. And then we also do fun stuff, so
Okay.
that helps. In Chicago, it's very hard to go out that
Okay.
much, but
Yeah.
summertime we do a lot of fun stuff.
Okay, that's
Wintertime
good.
we do snow stuff.
Yeah, that's good. Yeah. Uh, like since you mentioned mentors and all those things, can you please talk about your mentors and how do you do the mentoring for your people?
Yeah. I think mentoring is really what academic academia is all about. You really want to create the next generation of scientists. It's really less about what you accomplish yourself, but more about the legacy you leave behind.
Mm-hmm.
So mentoring is very important. I believe I've had phenomenal mentors in my life, and I am where I am because of my mentors. Starting from my mentors in school to my mentor in IIT Delhi, he was my he's my role model. Um, B. Jairam. He's a chemist in IIT Delhi, computational chemist, and he really sort of showed me how to be a good mentor. After that,
Okay.
Eric Cool, my graduate school mentor, same thing. What I learned about leaving people alone, letting them learn from their mistakes, letting them do whatever they want to do, what is in their interest. That's what I learned from Eric. And then subsequently Milton Warner at Rockefeller and Amin Arnaud at Mass General. So I've had phenomenal mentors, and I hope all the good things that I have learned from them, I'm imparting on my people. And really, in the future will tell how good a mentor I have been.
Yeah, good. Yeah.
I've had I'm also proud to say I've had one of the things I do differently than a lot of other people is every summer I take about 3 students, high school or early undergrad students, to come and get lab experience in my lab, and I believe
Mhm.
that that's a way to get very young people sort of exposure to science, lab science,
Uh-huh.
logical thinking, how lab experiments are done, how things are thought out. So that's something that we bring in. It really helps the lab because we get this really new energy.
Uh-huh.
Young kids who really wanna, they are there because they wanna be there, not
Well,
because their mom or dad said, "Oh, hey, go there." No.
yeah.
So that has helped a lot.
Okay. And talk about some of the projects going on in your lab.
So I have really two main streams of research in my lab. One
Mhm.
stream is looking at immune system, trying to understand how does our immune system work, and
Mhm.
then regulating it with new compounds, with discovering new drug-like molecules, therapeutic molecules that can regulate our immune system, and then a variety of diseases that go in that realm. And then the second part of the lab really is focused on kidney diseases, and there also we look at interplay of immune system and kidney system. So those really are two main areas of research. Underlying theme is drug discovery. We want to make new medicines.
Okay. Like I have seen some of your publications recently in Nature Communications and all. So you worked on lupus, lupus nephritis and all. Talk to us about a little bit about lupus. What lupus is? How does it come about? And what kind of autoimmune disease it is?
Yeah, good question, Sadashif. So, I'm going to backtrack a little bit. So, we really work on innate immune system. Innate immune
Okay.
system in the body really is neutrophils, macrophages, monocytes, cells that are essential for guarding us against infections, helping heal wounds. And it's these cells that are really the first responders whenever something happens in the body. And in majority of in autoimmune diseases, lupus being one such disease, in a majority of autoimmune diseases, these cells become overactive. And the last 15 to 18 years, what we have done is really try to understand mechanistically how do these cells work, how do they transit from bone marrow, go into through the blood vessels, and go to various parts of the body to do the job they're doing. And then where does something bad happen? What is a bad trigger, or how can you control a bad trigger that could lead to a chronic inflammation, which would essentially become an autoimmune disease? So really, that's sort of the underlying theme. The protein we study is a big surface receptor on these cells. It's one of the most highly expressed surface proteins. It's an integrin. So we are really using that as a handle to study function of these cells.
Okay.
Coming back to lupus, lupus is a chronic autoimmune disease. It is right now absolutely in need of new medicine. There has not been any effective new therapy developed for lupus in the last 40 plus years. It's a very difficult disease to treat. And it's complicated. The disease itself is, manifests mostly in women.
Hmm.
It's mostly in women of childbearing age, and it presents initially just as a fatigue or a joint ache or a joint pain, and so it's hard to diagnose initially until more blood tests and stuff are done. So it's a disease that's in a big need, and we got interested in lupus because the cell type that we study and the protein that we study, those two are critical factors driving this disease. So
Okay.
we figured that we can perhaps understand better and perhaps make an impact.
Okay. And going back to the immunity part of it, like where various innate immune cells, like you mentioned about macrophages and all. So, what triggers them to move from the bone marrow to wherever they are going? What is the trigger that happens?
Yeah. So typically, in our body, the bone marrow is the factory that makes all these immune cells, and these immune cells have a half-life in the blood. So they are continuously made and replenished in the blood, and that half-life typically for neutrophils is about 10 to 12 days. For macrophages, it's about a month, 20 to 30 days, and then they get recycled. And so they're continuously being made and sent as sentry in the blood, and then they go into the various tissues to really keep scanning for any infections or any bacteria, any viruses. But in the context of lupus, it seems that a number of patients, a number of people who go on to develop lupus or its complications, have a genetic predisposition which makes these cells prone to be overactive just at the smallest trigger. So somebody may get a viral infection, and it may just trigger this autoimmune response, an over or an excessive response from the immune system. Some people may get this because of some medications that they are taking. It's really not clear right now what triggers lupus, and that's a big area of research going on right now, so that people can sort of avoid those triggers. But it's not really clear at all.
Not, people don't know still what happens, and lupus, like I remember, if I read it right, there are some as you said, it is a genetic disease, and there are variations of these genetic diseases. So can you please explain where exactly, what is the chromosome, and little bit about the genetic part of it?
Sure. So so actually, it's funny, and I was teaching that to my daughter, high school. She's a freshman in high school yesterday. So
Okay.
it's timely. um, so chromosomes are really our storage for cells. Chromosomes are what keeps all the information in a cell, and chromosomes in human body, there are 23 pairs of chromosomes that encode for all the genes, all the properties that we inherit, all our traits. And there are more than 20,000 genes in our body, as you know. And again, different cell types have different proteins that are overexpressed or underexpressed in them, that sort of gives them their phenotype, gives them their identity. And same for people. Our chromosomes encode for genes that ultimately dictate a variety of our traits. This decade, I believe, has been revolutionary for science in this field, because all the new technologies and tools that have been developed to really understand what are different genes, what do different genes do, what do different genes do in different cell types, what do different genes do in different organisms, humans, chimpanzees, mouse, rat, dogs, So it's a very exciting time in a way to be studying genetics of a disease, and lupus being partly inherited or predisposed due to mutations in genes. This is really, I think it's an exciting time to be doing that research.
Okay. And like as you mentioned, there is these cells are activated and going to different, like it's autoimmune disease lupus. So like that means like antibodies are generated. So can you please mechanistically explain what happens when there is infection, these macrophages, neutrophils rush to the different places, and the autoantibodies are generated? What happens basically?
Yeah. So what is currently understood is due to some trigger. So first of all, lupus as a disease isn't always in a what you call flare. So people with lupus lead very normal life most of the time. It's only certain periods in their life that there is some trigger that leads to this overactivity of immune system, and that activity period is called the flare. And at the time of that flare, things just go haywire. And presentation wise, then the people have fatigue, as I mentioned, fatigue and joint pain, and it could also manifest in skin inflammation and in renal inflammation, cardiovascular damage, brain damage, things like that. So what typically people understand is at the time of a flare, there is a lot of neutrophil activity that leads to shedding of internal DNA of neutrophils in the blood. And typically in the blood of in human blood, you don't really find pieces of DNA. So when the pieces of DNA get exposed in human blood, other immune cells, B cells that make antibody and protect our body from pathogens, they see that as a foreign thing. They think it's a virus. They think it's some infection that's trying to invade the body. So they detect that DNA and start making autoantibodies against the DNA. And
Okay.
that anti-DNA autoantibody is a primary signature in blood of lupus patients.
Okay.
And that's something that that gets that gets picked up as a serological marker in patients. So you have overactivation of neutrophils. Neutrophils degrade, expose DNA out of them. That DNA leads to production of autoantibodies against the human neutrophilic DNA. And then when there is a huge abundance of these antibodies, they sometimes cluster, form complexes, and then these formed essentially beads of proteins are then going through the blood. Blood gets filtered through the kidney, and a lot of times these things get stuck in the kidney, for example, and thus induce renal damage that is dependent on lupus. So lupus nephritis, for example.
Okay. And in terms of the mechanistic action, these neutrophils, macrophages, so they have some proteins, they respond to the other cells. Like, can you please explain that part of it? What are the proteins get expressed? How does it happen?
Yeah. So the main protein on neutrophils is this receptor that we are very interested in the lab. It's called CD11B, which forms a receptor called MAC1. So CD11B or MAC1 on neutrophils. Neutrophils are essentially like spherical tennis balls floating in a bloodstream. Essentially, think of it as a bloodstream being a pipe and the blood being the water in the pipe. Then these beads, tiny beads of neutrophils are just going through the blood. And then whenever there is an inflammation, this tennis ball, where the fur on the tennis ball is the receptors. These receptors are this integrin CD11B. These fur balls stick to wherever the infection or inflammation is with that fur like Velcro. So essentially, this protein targets neutrophils or macrophages to the sites of infection or inflammation in a normal setting. And in the context of an autoimmune disease like lupus, this fur becomes overly sticky, for example, or overly active, that produces more damaging response from these cells. Does that make sense?
Yeah. Yeah. And you talked about CD11B. So they are supposed to be the integrins, right? Integrin
Yes,
receptor.
yes.
Can you please talk about a little bit about integrins and their function?
Yeah. So now again, that's a good way to segue to sort of talk about integrins. So stepping back, integrins. Every cell in the body has a type of receptor called integrin. Integrins are the proteins that cells use to basically um, think about or interact with their environment. So cells use integrins to interact with another cell, or cells use integrins to interact with the matrix or another layer of proteins. So integrins are these key essential receptors on every cell type. There are 18 different types of integrins in our body, and different cells express these different integrins. The integrin in neutrophils and macrophages is composed of CD11B with its binding partner CD18. And so this integrin, also known as MAC1, really is essential for function of these cells. These cells express the integrin, and this integrin makes helps them migrate away from bone marrow into the blood, from blood into the tissue. It also regulates by signaling, by sensing the outside. It also helps signal inside the cell and generate a lot of responses that come from these cells. So one other thing I wanted to just say about integrins is really, the name comes, name derives from its being an integrator of signals. So thus, integrins. It's one receptor that integrates outside, senses outside, integrates the signal, and tells the inside of the cell what to do. It also integrates signals from inside of the cell and tells the outside what to do in terms of the cellular response.
So can you talk about those signals, like mention them, like TLR4 and all those things, like how does it like these integrins binds to the cells on the surface, right? So
Yeah.
other cells, maybe endothelial cells or other cells. So what is that interaction is about, and what is the signaling happening, and what that means to the cell, like whether they are
Yeah.
binding, migrating, little bit more explanation about the signaling aspect.
So typically, for cells that are sticky or adherent cells, like endothelial cells, their integrins are required for their viability. Their integrins are required for this adhesion-mediated viability of cells. That's one of the most common things that is known about integrins. So in those cells, the cells use this integrin to bind to their neighboring cell or bind to a matrix, and that binding tells the cell, okay, stay alive. In the case of neutrophils, the integrin that we study, CD11B or MAC1, that neutrophils are not sticky in a way that they are circulating. So they're always circulating in the blood. The integrin is in a conformation that does not make the cell sticky. But and in response to certain injuries, certain cytokine signals, for example, from injured sites, certain concentration of bacteria, certain bacterial products or viral products that then interact with a different receptor on the same cell, in this case, toll-like receptors or TLRs. So bacterial products or other products would then bind to TLR on the same cell. TLR activation then tells integrin to activate and make cells sticky. Stop. There is some danger there. So the neutrophil, instead of just zipping by in the blood, would then suddenly come to a stop to figure out what is going on near this region where there is an increased density of TLR ligands. And once the neutrophil sticks, it then secretes more things to tell other neighboring neutrophils, okay, you guys, stop. We got to look at, survey what's going on here. And thus there's an accumulation of these immune cells, neutrophils, or monocyte macrophages in a region in the body near a blood vessel where there may be increased density of TLR ligands. And that leads to this adhesion, and that may also ultimately lead to a response from the neutrophil, in which case neutrophil just sheds its DNA and thus activates additional signaling in the body.
Okay.
So in a way, now bacterial sensing by TLRs is then adding a signaling via integrins to the cell to tell what to do in terms of adhesion. There is a second response that also happens in the body, which is when the TLRs and the integrins are both activated and adherent, then they tell the cell inside of the cell to start making a lot of pro-inflammatory molecules, IL-1 beta, IL-6, TNF alpha. These are small soluble factors that are then secreted out of the cell and tell the surrounding cells of any kind, to really be over vigilant, and thus this you create this pro-inflammatory environment. So TLR activation, 11B activation at the same time, and a lot of cell adhesion, that leads to this pro-inflammatory phenotype. Does that make sense?
Yeah, yeah. So once it is done, like how does it cure? Like for example, in lupus, what happens once the auto antibodies are generated? They go in the lupus nephritis. They block this thing. So how do you take care of it, and what happens, and what is your work on some of the agonist for these
Yeah.
anti integrins?
So, so knowing what I just told you, which is people thought CD11B and TLR mediated cell adhesion is essential for the activity of these cells, the pro-inflammatory activity of these cells, and this overactivation leads to these autoimmune diseases. In the past, people had said, okay, you know what, let's just block CD11B, and if you block it, then this response will not happen, and thus you would have a reduction in autoimmune disease. However, a lot of research suggested mixed phenotype, that in many cases blocking CD11B was not enough to overcome this pro-inflammatory signaling change that the cell is seeing. So we reasoned many years ago that, well, let's see if we just switch the CD11B from inactive to active conformation without really doing anything else. So in that setting, we wanted to find out whether it's going to increase disease or decrease disease, not really knowing what would happen. So we developed we developed a whole series of small molecules that bind to a certain allosteric pocket in CD11B. They bind to CD11B, induce a small conformational switch, and that conformational switch induces or converts CD11B into a pro-adhesive phenotype, meaning if there is a high density of ligands, the cells might stick. Otherwise, the cells just keep zipping by. So our CD11B agonists, what very surprisingly we found was they actually act as a negative feedback loop on TLR signaling in this immune context, in this autoimmune context. In circulating cells, these neutrophils and macrophages and monocytes, in those, usually by getting TLR activation, produce more pro-inflammatory signaling, but when CD11B is activated, they suppress TLR signaling, thus reducing autoimmune disease in general. And we found now in several mouse models where activation of CD11B by administering our compound and activating CD11B, they can suppress this autoimmune disease. So we are very excited about it. It, I think, could be a new way to develop therapeutics against lupus and other autoimmune diseases.
So you mean to say when these are activated, still they bind to TLR4, but whatever the signal, like what you're talking about, interleukins coming out, they are not there anymore, or less number of.
Correct. So typically TLR, toll-like receptor binding to its ligand, like LPS or other molecules, induces this pro-inflammatory signaling in the cells. And what we found is activation of CD11B really acts as a negative feedback loop on that signaling mechanism. So it really suppresses generation of this, IL-1 beta, IL-6, TNF, these types of inflammatory markers in this context.
So is it by binding to the same TLR4 receptor, it activates certain different signaling pathway, or it does it through some other pathway?
Yeah.
Or it is just intracellular? What is the, like, I want to know a little bit more
Yeah.
about if it is known already.
Yeah. Good question. So we actually published this recently in our JCI paper where we showed that typically what happens, a TLR signaling, TLR binding to LPS induces or activates MyD88. That's its adapter protein in the cytoplasmic domain. So TLR binds to LPS, activates MyD88. MyD88 then activates a whole bunch of signaling kinases that ultimately lead to activation of NF kappa B. And that NF kappa B, then being a transcription factor, goes to the nucleus and induces an overexpression of pro-inflammatory genes. The second thing that TLR does, which we discovered, was TLR activation also induces AKT phosphorylation, and that results in degradation of a second transcription factor called FOXO3, and that results in generation of more type 1 interferons, interferon 1, interferon 2. So it's just this TLR dependent pro-inflammatory NF kappa B signaling and TLR dependent pro-inflammatory interferon signaling both happens in cells in the absence of CD11B engagement, the way I was talking about. And what we
Okay.
found is by activating CD11B, it really induces a second set of kinases, SRC and SYK, which phosphorylate ubiquitinase. That ubiquitinase MyD88 removes it from the signaling pathway, and that just changes all downstream signaling. And that downstream change is reduced NF kappa B activation and enhanced FOXO3 transit to the nucleus, thus suppressing interferon generation. So both reduction of NF kappa B and reduction of interferon signaling in this context.
Okay. So now recently you had some good results on cancer, right?
Right.
Like you are connecting the same immunity to the cancer and decreasing tumor growth and all those things. Can you please talk about that a little bit?
Yes, certainly. So this is our recent paper in Nature Communications. Again, work with Judy Warner's lab at UCSD, where we applied the same hypothesis in the context of a tumor microenvironment. And overall, the idea was we want so just switching gear a little bit. In the autoimmune disease, the cells in circulation play a big role, and there they generate these pro-inflammatory signaling molecules in circulation. In the context of tumor, a lot of action happens in the tumor microenvironment itself, where it is known that the tumor microenvironment recruits macrophages and other myeloid cells from circulation, and there they go and they completely adopt a pro-tumor phenotype, where they start helping tumor grow by inducing neoangiogenesis, as well as suppressing T cell infiltration that could kill the tumor cells. So if this were the tumor, these macrophages go and form a barrier around the tumor, and that barrier prevents T cells from infiltrating as a physical barrier as well. So there are many things these macrophages do. And again, CD11B being a key highly expressed receptor on these cells, we wanted to see if that would have an impact on this sort of tumor growth phenomena. And again, not very surprisingly now, but surprisingly overall, we found that CD11B activation kept most of the myeloid cells in circulation. It prevented them from going from blood vessel out into the tumor sort of microenvironment. So you could remove this physical barrier that was preventing T cells from coming in and getting to the tumor. And that has been a very satisfying sort of research project as well, where we sort of said, okay, remove this layer, see what happens to the tumor, and then we very, very quickly saw in many different tumor types that the T cells are now able to come in and recognize the tumor and get rid of it.
So how, like you have mentioned most of the cancer stuff and all, what is your path forward. So what are you looking
Yeah.
at?
So one thing I forgot to mention at the before the start of this is I do have a conflict of interest on this research in general, which is I do have a startup that is trying to take these discoveries into clinic. And again, my goal really is to find new therapies for people. So my startup, Adhere Pharmaceuticals Inc., is trying to develop these discoveries, licensed from all these research universities where my lab has been. So the path forward is we want to take these compounds. We have now tested them in a lot of tox studies in animal models, and we found them to be very clean. They are non-toxic small molecules. Uh, and they are bioavailable orally. So we want to now take this same idea, activate CD11B using an allosteric agonist, and test this idea in patients with a variety of these immune disorders. I'm lumping cancer in immune disorders because it is an immune disorder. So testing our CD11B activation as an idea in these treating immune disorders in patients. We are hoping to get to clinic very soon with guidance from FDA, and then really test this idea in patients, and hopefully we make a difference.
So thank you very much. So it's really fruitful. Like I learned a lot myself trying to untangle the thing because I'm not an immunologist per se. So it helped me to understand a little bit more in detail on what is happening and all. And I hope the viewers will learn a little bit more about the immunology and the work done by you and from your lab. So thank you very much for helping me understand the lupus and a little bit about the immunity. So if you have something new, you are free to come again and talk about your new work. Uh, so thank you so much.
Great. Appreciate that, Sadashiv. Appreciate it very much. Thank you.
Thank you.